Hi, this is Kavita Kundra and we are covering today the last part of Lipid and Protein chapter, part 4 of MSc DFSM. Peptides. What are peptides? Peptides are formed by linking amino acid by peptide bond formation that involves carboxylic group and amino acid with the elimination of water molecule. Polypeptide and protein are the chain of amino acid held together by peptide bond. When two amino acids join together, they form dipeptide. When three amino acids join, they form tripeptide, and so on. When more than 10 amino acids join, they form oligopeptide. Each amino acid begin with amino group and end with carboxylic group. Physiological, Physiologically important amino acids are glutathione. It is a tripeptide made up of glutamic acid, cysteine, and glycine. This is an important part of oxidation reduction reaction. Oxytocin and vasopressin, they have 8 amino acid and they help in injection of milk and reabsorption in kidney. Angiotensin. Angiotensin 1 and angiotensin 2 have 10 and 8 amino acid respectively and has slight effect on blood parameters. Insulin, it has 51 amino acids and it has two polypeptide chains linked together by two SOS bridges. It helps in the utilization of sugar by cell. Proteins, these are the dehydration products of amino acid with each amino acid residue joined to its neighbor by specific covalent bond. Classification of proteins, one is simple protein, another one is conjugated protein, and third one is derived. What are simple proteins? Proteins that carry only proteins and no other compounds are called simple protein. Example, albumin and globulin. Conjugated proteins, these are the proteins, they have non-protein part as well with the protein part called nucleoprotein or lipoprotein. Derived proteins are the proteins formed from either protein or conjugated protein due to unfolding of tertiary structure or cleavage of peptide bond. Example, primary derivatives are proteins and metaproteins. Secondary proteins are peptones and peptides. Structure of proteins. There are four levels of structure. One is primary, secondary, tertiary and quaternary. In primary structure, it refers to the linear arrangement of amino acid residue in given polypeptide chain linked through the peptide bond, and the shortest peptide bond has 20 to 100 amino acids secreting. Most of the proteins have 100 to 150 amino acid. Rarely they have 1000 amino acid. The number and sequence of amino acid determine the specificity of protein, and slight disturbance can react a new protein. Example, hemoglobin, where glutamic acid is replaced by valine at position number 6 of hemoglobin. It changes the hemoglobin and creates this disease called sickle cell anemia. Secondary structure, it refers to the three-dimensional arrangement of various atoms of protein, and the polypeptide is folded and secondary conformation is stabilized due to the binding forces. Hydrogen bond, disulfide bond, ionic bond and hydrophobic bond are formed. Some protein take the shape of coil like alpha helix, alpha keratin, and some protein possess sheet, beta plated sheet, example beta keratin or collagen. Tertiary structure, this term refer to the tendency of polypeptide chain to undergo extensive coiling or folding and produce a complex and rigid structure stabilized by intermolecular bond like hydrogen, ionic, hydrophobic, and it attains globular and ellipsoidal conformation. Quaternary structure. Many proteins like enzymes consist of peptide chain linked by disulfide bond. Hemoglobin consists of 4 polypeptide chain, 2 alpha chain and 2 beta chain bound to. Then is physiochemical properties of protein. Isoelectric pH. Many ionizable groups are present in the protein molecule. Depending upon pH, some act as proton acceptor and some as proton donor. Thus, protein is amphoteric compounds at specific pH. It acts as dipolar ion or Zwitter ion. This pH is called isoelectric pH. Solubility. Globular protein are generally more soluble in aqueous medium as compared to keratin or fibrous one. Solubility depends upon pH, nature of solvent, temperature. Precipitation. Isoelectric pH, as they do not have any charge at isoelectric pH, amino acid become precipitated. Salting out in aqueous medium, protein can be precipitated by adding TCA, trichloroacetic acid, or PCA, perchloric acid. Salts of heavy metals like like phosphomolybdic acid and phosphotungstic acid are used for precipitation. This process is called salting out. Concentrating solution like MgSO4 or Na2SO4 also used for precipitation. Action of non-polar organic solvents. A non-polar solvent like chloroform enhances the electrostatic attraction between ion of protein and result in precipitation. Chemical properties of protein are due to the side chain. Sakaguchi reaction. The arginine side chain, each containing a guanine group, react with a naphthol in presence of sodium hypochlorite to produce red color. Hobson-Cole reaction, in which tryptophan side chain being indole can react with glycoxylic acid in presence of concentrated sulfuric acid to form purple color. Third reaction is Millian reaction, in which tyrosine side chain, each possessing a phenolic group, undergo variety of reaction. If it is treated with mercuric sulfate and sodium nitrate, red complex is used. These reactions are used in quantitative analysis. Structure and classification of nucleic acid. What are nucleic acid? These are the polymers of nucleotide. Nucleotide are the nucleoside phosphate. Nucleoside is composed of nitrogenous base and pentose sugar. Nitrogenous base can be further classified into purines and pyrimidines. In purines, adenine and guanine is there, and pyrimidines, cytosine, uracil and thymine comes under pyrimidine. Pentose sugar is also two types, ribose sugar and deoxyribose sugar. When phosphate group, pentose sugar and base combine, it form nucleoside. When the sugar present is ribose sugar, it is called ribonucleotide, and if the sugar present is deoxyribose, it is called deoxyribonucleotide. AMP is naturally occurring nucleotide, and during cellular metabolism, AMP with ADP, adenosine 5 diphosphate, and adenosine 5 triphosphate means ATP plays very important role in the utilization of. What are the comparisons of RNA and DNA? What is RNA? RNA is ribonucleic acid. What is DNA? DNA is deoxyribonucleic acid. RNA has ribose sugar, DNA has deoxyribose sugar. It has uracil in presence of thymine and others are same. DNA contains 4 types of nitrogenous base: adenine, thymine, cytosine, guanine. RNA is single-stranded as made as complementary strand of one of the 2 chain of DNA, and in DNA there are 2 polynucleotide chain coiled around central helix. RNA is small, DNA is long. There are 3 types of RNA. mRNA means messenger RNA, tRNA means transfer RNA, rRNA means ribosomal RNA. They all have specific function during protein synthesis. In DNA, the chain is held together by hydrogen bond between the bases of opposite chain. So base pairing is very important. Adenine, A always pairs with T, thymine, and guanine always pairs with cytosine. Thank you so much. I hope you understand. This is very easy chapter. And if you have any questions, you can ask me in comments. And I hope you will clear your examinations, and otherwise you will enhance your knowledge about nutrition and biochemistry and DNA, RNA. These are very interesting topics if you go through these. And I hope you will do better. And stay safe, stay protected. Bye bye. Take care.